Hi there. I currently work for the East Foundation in South Texas, but I'm going to talk today about actually my undergraduate research looking at parasitic infection in African equids. I did this research as part of the Princeton University Zebra Research Project at the Impala Research Center in Kenya. The PI on the project is Dr. Dan Rubenstein. I worked with him and his then PhD student, Dr. Kaya Tombak. We were actually interested in using the theory of island biogeography to think about parasitology. So we know that this theory says that the number of species that are found on an island is a product of both the colonization rate of species coming onto the island, as well as the extinction of those species off the island. And there's different factors that can elevate or decrease both of those rates. When you're thinking about parasitology, you can conceptualize actually a host animal as sort of an island that's being colonized or infected by parasites. And that host also has the opportunity to try to extinct those parasites through an immune response. And so a host's ultimate parasite load, it's going to be a factor of both that infection risk with parasites and the immune response against parasites. So we wanted to look at both of these factors in this study of parasitic infection in African equids. The equids that I studied were co-occurring in Kenya. They included plains zebras and Grevy's zebras. Grevy's zebras you might not be familiar with. They're an endangered species and they're a little bit different than plain zebras in that they have an adaptation to arid environments. Specifically, other than the lactating females, Grevy zebras do not have to drink water every day. So they're able to range farther out from water points and access different parts of an ecosystem. Plain zebras don't have that adaptation, so they do need to drink water every day and be able to every day get to water. The other type of equid that you have co-occurring with these zebras is the domestic donkey. Historically kept as a beast of burden, donkeys are put overnight into corrals with other livestock, and then each day they're brought out with cattle to go graze on a rangeland. The parasite that I studied is the strongyle. These are nematodes from a variety of genera that are specific to equids. Strongyles inhabit the gastrointestinal system and they are bloodsuckers. Now, although the strongyle infection rate might not often be high enough to cause pathology, if it does get to be an intense enough infection, you could see intestinal problems, weight loss, and that. The way that strongyles are transmitted is through a fecal oral route. So you have adult worms living in an equid's gut that produce eggs that are shed through the feces. Those eggs eventually hatch into larvae that develop through some different stages and ultimately crawl up onto the grass where they can be eaten by an equid. And then that larvae develops into an adult worm in the gut. Because this is a fecal oral transmission route, an equid's infection risk, it really depends on its exposure to dung that has eggs and larvae in it. I did my study in the summers of 2017 and 18, and I found my species at the Impala Research Center. It's in central Kenya in East Africa. This is about a 40,000 acre savanna ecosystem, and most of my work was done on these colored loops in the southern part of the property. So every day I would drive these different loops looking for groups of zebras. I would observe zebras and wait for individuals to drop some dung. I would note which individual that was and then go out there and pick up that sample. I'd bring my samples back to the lab and I was quantifying two metrics. First, for the parasite load, I was looking at fecal egg counts, really the number of eggs shed per gram of dung. Of course, egg load isn't going to be a perfect indicator of parasite burden, but it gives you a sense of maybe how many adult worms are in that gut, and it's also non-invasive to measure. So I would count eggs under the slide using the McMaster technique. In the summer of 2018, I was also able to measure immunoglobulin A or IgA levels. So IgA, it's kind of the primary intestinal antibody that mammals have, and it's kind of considered the first line of defense in the gut. So I wanted to look at how this was relating to the strongyle infection. IgA was measured using ELISA assays and staining using those dung samples. So getting into some of my results here, I want to look first at that colonization or infection side. And one of the first things I saw is that for each species, the parasite load, that egg count was significantly higher in 2018 compared to 2017. And when you look at how this landscape looked across these 2 different years, you can see it's very green and lush in 2018. 2017, it had half as much rainfall in the months leading up to and including my study, and it was very brown and barren. Now we know that strongyle larvae, they survive and they develop the best when the environment is moist and when they are provided with shade. So in 2017, you had all that bare ground. Likely these strongyle were getting desiccated by the sun. But in 2018, with all that vegetation, they were shaded and they were able to survive, develop, and ultimately infect those equids at higher rates. So that increased exposure to more alive larvae probably caused this increased parasite burden. Looking at the same graph here, if we start focusing now on the differences between the 3 different species, there was a trend in my study and in other studies as well that Grevy zebras have lower strongyle loads than plain zebras or donkeys. And we think that this also relates into that infection risk or colonization risk. Remember that Grevy zebras are not having to drink water every day due to their adaptations, so they can range farther from water. It's been shown that zebra dung is most common and highest densities near water points, and it decreases with distance from water. So Grevy zebras far from water, they're being exposed to less dung and less parasites compared to these other species like plain zebras. They're always staying within a day of water. And donkeys, which spend the night in their corral every time and probably often go out to graze on range that has their dung from previous days or weeks. So Grevy zebra's ecology might be giving them lower parasite exposure and lower parasite loads. In addition to the differences between the species, you can also see differences just between individuals within a species. I actually had about 11 Grevy zebras that I repeatedly sampled in 2018, repeatedly observed, and I saw that their parasite load throughout the summer differed by individual, and thought this could also be explained through that colonization or infection risk and really their use of high activity areas that have a lot of dung. I already mentioned that places that have water points have higher dung densities due to the animals coming in to drink. Also wanted to look at glades as high activity areas. So glades are areas that used to have a temporary livestock corral. That corral's been moved, so we're not getting that area trampled every day by livestock, but all they're dung there is this amazing nutrition that causes basically a lawn of green grass to form. And that can be very attractive to these grazing zebras who might have high activity at that glade, have a lot of dung there, and then there could be higher parasite exposure. So I made kind of a crude linear model for 11 Grevy zebras, trying to explain their parasite load from the distance of their observed home range to a water point, as well as how often I was observing them in a glade versus not, and sort of how high activity that glade was. And it really showed that both of these factors matter for exposure to parasites and that ultimate parasite load. And just diving a little bit deeper into this idea of glades, a lot of the individuals that I repeatedly observed and could kind of see a home range for, which you see in these colored polygons, they were somewhat touching, most of them were touching a glade circled in that green. And that really shows you that human modification of the landscape through livestock production, it's altering the way animals behave and move. That impacts their exposure to parasites and ultimately their parasite burden. Shifting gears a little bit, I now need to talk about that extinction or immune response side of the story. And I'm do that first through the story of this individual zebra. I named him Chip because of that chipped ear, and I sampled him over a dozen times throughout summer 2018 until I was able to track his IgA levels there in the red and the egg count in the blue over a lot of different days and sometimes even subsequent days. The first thing you see in this graph is that IgA level in the red is bouncing up and down. It's not staying constant. And this seems to be suggesting that IgA is like turning on and being activated at certain times and then turning back off. And that suggests that this is an inducible defense. It's being activated in response to some need compared to staying at that same level and being a constitutive defense that stays the same every day. You also notice in this graph that when you do have that peak in IgA level, we also in subsequent days see that the strongyle egg load is lower. So that's suggesting that maybe this IgA is in there actually resisting these parasites, reducing the parasite load by, say, fighting back against those adult worms, fighting their fecundity and their production of eggs. So knowing that IgA maybe is a resistance defense to lower the parasite load is interesting because it was also seen that Grebe zebras produce higher IgA levels than plain zebras or donkeys. You'll recall that Grebe zebras always trend to having lower egg counts, and so maybe that's a product not just of that lower colonization or infection risk due to their movements getting away from water, but also their production of higher levels of this IgA that seems like maybe it can reduce the parasite load. And in terms of thinking which is more important, the infection risk or the immune response, well, one area to study that could be in hybrid zebras. These are ones where the father is a Grebe zebra, the mother is a plain zebra. They live with the plain zebras and go and drink water with those groups every day. And so you can look at maybe untangling that infection risk of drinking water with the plain zebras versus having some of that maybe immune response and physiology of a Grebe zebra. So I said that my study was going to have some key lessons in immune ecology. To review those on the colonization or infection with parasites side, I saw that environmental conditions impact parasite load, likely through that exposure. In a rainy year, we had more exposure and infection with these parasites that are transmitted fecal-oral routes. Also saw that the ecology of species as well as the behaviors of different individuals impact their exposure to parasites and their parasite loads. And that when humans modify the landscapes, that changes the way wildlife moves, gets exposed to parasites, and has their infection load. On the extinction side, we saw that IgA seems to be being activated and turning on in response to parasite burden, and that indeed we can have immune responses actually reducing that parasite load. And so all in all, it is important to look at both that colonization infection side as well as the extinction or immune response side when looking at parasitic infection in wildlife. My research was used for a couple publications by Tombach et al., so definitely encourage you to take a look at those if you're interested. Thank you so much.